Okay, let's take a look at the pyruvate dehydrogenase enzyme and the activity that it performs in the mitochondria. The first consideration we have to make is, how do we get pyruvate into the mitochondria? Remember, the mitochondria has a double membrane. This is the outer membrane. This is the inner membrane. And each membrane has a very different property. This is the inner or intermembrane space. Each membrane has a different property because of the regulated transport that is necessary for the inner mitochondrial membrane. So the outer mitochondrial membrane has a level of porosity that molecules less than 6,000 molecular weight, or Daltons, can pass without hindrance, and no transporter is needed. Now the pyruvate has to be transported across the inner mitochondrial membrane. So as we saw in our previous video, glucose is oxidized down to pyruvate in the glycolytic pathway. So now pyruvate can pass through freely the outer mitochondrial membrane by diffusion, but once it reaches the inner membrane space, there has to be regulated transport. For this purpose, we have a symporter which is going to transport pyruvate into the mitochondria, as well as transport protons from the inner membrane space into the matrix of the mitochondria. This proton transport is due to a proton gradient that has a very large proton concentration on the outside of the inner membrane and a small proton concentration on the inside. We'll talk more about this. This is established by the process of oxidative phosphorylation, electron transport chain. So now once we have the pyruvate transported as cotransport with protons, now we need to process the pyruvate into acetyl-CoA. So here's our pyruvate. Here's our acetyl-CoA. This step is necessary in order to take the acetyl-CoA and place this into the TCA cycle. The enzyme that does this reaction is the topic of this video, pyruvate dehydrogenase. Now the pyruvate dehydrogenase enzyme is a very large complex. It's consistent of 3 complexes of enzymes. They are designated E1, E2, and E3. But the most important part of this is the cofactors that are involved. One cofactor is abbreviated TPP, which is thiamine pyrophosphate. The importance of this is derived from vitamin B1. So individuals with a deficiency in vitamins, especially the B complex, B1, will have fatigue because they're not able to perform energy production. The other 2 factors involved are lipoate and FAD, because this is going to be a redox reaction. Now, we're going to need a cofactor coming in here, which is coenzyme A, abbreviated CoAsh, to form the acetyl-CoA. Notice that the 3-carbon pyruvate has been transformed into a 2-carbon acetyl-CoA. So this is going to be also releasing CO2. And finally, this complex reaction is a redox reaction. So we will be converting NAD into NADH and harvesting some electrons during this process. Now this whole process is an oxidative decarboxylation. We'll see two more of these reactions in the TCA cycle. Now this enzyme is regulated by pyruvate kinase. The pyruvate kinase is controlled largely by product formation. So let's put the pyruvate kinase here. We'll write pyruvate dehydrogenase in the inactive form below, and pyruvate dehydrogenase in its active form above. Now the conversion of the active to inactive form by the kinase is going to be done by a phosphorylation reaction. And in this case, the pyruvate kinase will be using ATP, converting this to ADP, and phosphorylating the pyruvate dehydrogenase and activating it. For activation, we are going to need a dephosphorylation event, releasing inorganic phosphate, and this is the protein phosphatase function. So what controls the pyruvate kinase from doing this cycling activity of phosphorylation of the dehydrogenase enzyme are concentrations of acetyl-CoA, so the substrate of the pyruvate dehydrogenase will activate the kinase. The other product of this reaction is the NADH. NADH will also activate the kinase. If the kinase is active, it will take ATP and phosphorylate the dehydrogenase enzyme in activating it. And this is in response to buildup of product to slow the dehydrogenase enzyme down. Now to turn the pyruvate dehydrogenase enzyme on, we need to dephosphorylate, and that is going to be done by the phosphatase activity. This activity will be promoted by calcium. Calcium will also inhibit the kinase activity. By doing this, we have neural impulses stimulating skeletal muscle, causing depolarization, which leads to contraction. Now this contraction event here is a release of intracellular calcium stores. So the combined effect of calcium is to mediate the contraction event, but it's also to activate energy production. And this is to compensate for the decrease in ATP because of hydrolysis needed for contraction. So the activation of energy production is mediated through activating pyruvate dehydrogenase by converting it to its dephosphorylated active form by the pyruvate kinase enzyme, and the phosphatase activity being activated, pyruvate kinase being inhibited.